Protists have a wide array of ways that they get their food, and this is due to their evolutionary history. So let's take a look at some terms related to how they get their food, and then explore how they ended up with really diverse ways to access food. So phagotrophy is protists that are heterotrophic, or organisms that are heterotrophic, because they can ingest particles. So this could be pieces of other things, of other living things, or in some cases, it's entire cells that they're able to ingest. Osmotrophy is when they're heterotrophic, but they can only absorb small organic molecules. So this relies on broken down stuff in the environment. So they can't uptake a whole piece of an organism. They have to rely on small molecules floating around in the fluid from something getting broken down. So a dead cell that already broke down into its components, or sugar in the environment or something like that. Photoautotrophy refers to photosynthetic organisms. So they're able to use light to make their own food. And then this one is the one that's particularly unique to protists, mixotrophy. These are cells that are able to carry out photosynthesis, that is, they're autotrophic, and they're able to eat other things, either through phagotrophy or osmotrophy, depending on conditions or the cell type. So basically, they can do photosynthesis and they can eat. And so how cells get to be able to do both is due to some unique things that happened in protist evolutionary history. So this has to do with endosymbiosis. So we've talked about endosymbiosis with bacteria and the first eukaryotic cells. We're going to get into that a little bit more by distinguishing primary, secondary, and tertiary endosymbiosis and seeing how mixotrophy arose in protists. So we'll take a look first at primary endosymbiosis. And so this is the type that gave rise probably to chloroplasts. So you have a bacterial cell that was engulfed by another cell, in this case a eukaryotic cell, a protist. So this is the protist. This is the photosynthetic bacteria. So this cell wasn't able to do photosynthesis before. It takes in that cell, and when it takes in the cell, it absorbs it through its membrane. So when it goes across that membrane, it has to wrap the thing that it brings inside of itself in another layer of membrane. So to show you what I mean, let me use another color here. So this is going to be my protist, and my bacteria is going to be this little green cell. So when the bacteria is taken inside of the protist, it has to wrap the bacteria in its own membrane. So basically, when endosymbiosis happens, there's this chance that the membrane gets another, or the thing that went inside the cell gets another layer of membrane. So you have your, and so let me do this a little bit more. So inside of your bacteria, you may remember that we have thylakoids. So you end up with your thylakoids. This is what the green here is the inner membrane of the chloroplast, and this blue here is what becomes the outer membrane of the chloroplast. So if you think back to what you learned about the chloroplast, it has two membranes plus the thylakoid. And so it gets the thylakoid membrane, the inner membrane, and the outer membrane due to its evolutionary history of having arose through primary endosymbiosis. Okay, so that's what we have focused on before. However, secondary endosymbiosis can happen. So basically, imagine if this protist then gets eaten. Okay, so keep in mind that this protist ate the bacteria, but now we're imagining what would happen if this protist got eaten. And just take a look at this guy here, because we'll see it in the next picture as well. So this is that another cell that had engulfed a bacteria. So this is now a eukaryotic cell. in this case, a green algae, for example. It could also be a red algae, which we haven't talked about yet, so we'll just ignore that one for now. But it can be a eukaryotic cell that has a chloroplast. And where did that chloroplast come from? Remember, it was once a bacterial cell. Okay, so now this same type of cell is able to engulf this food. And so it can either engulf this one or it can engulf this one. Okay, and what happens is just like before, it's taking that and putting another layer of membrane around it. So remember, I had my cell here. I had my cyanobacteria that got engulfed. There's its thylakoids. And remember, when it got uptake by this other cell, it had another layer around it. Well, let's use another color. Let's imagine this cell has yellow. Well, when it ate this cell, it put another layer of membrane around it. Okay, so every time we have endosymbiosis, there's the potential for another membrane layer to end up on the cell, or around the chloroplast rather. So here's my original chloroplast that's going to end up with multiple membranes around it. If secondary endosymbiosis happens, it has more membranes than primary endosymbiosis. So this is getting at, okay, how do they know this, right? How do they know that secondary endosymbiosis happened? Well, one of the ways that we know it is by seeing additional layers of membranes than you would have predicted. And so this can happen a third time, tertiary endosymbiosis. So imagine if this cell now got eaten by another cell type. So here's another cell type engulfing this other type of cell. So let me go back to my drawing. My drawing's getting pretty complicated here. So I had that original cell. There's my bacteria. It got wrapped in this membrane. Okay, then I had my second type of cell. It wrapped this in membrane inside of that cell. And now I have another type of cell that's going to eat this. So again, you get additional layers of membrane when tertiary endosymbiosis happened. And so cells that have this oftentimes are mixotrophic because they had the ability to engulf other cells, but then they also acquired a chloroplast. Okay, and so that's what makes them mixotrophic. They had already evolved the ability to ingest other cells when they acquired a chloroplast through tertiary endosymbiosis. So now they can do both photosynthesis and they can eat things. And again, part of how we can support this with evidence is that we see additional layers of membrane. So primary endosymbiosis, to summarize, is when a cell engulfs one cell and retains it, in the case of how chloroplasts first evolved. Secondary endosymbiosis is when a cell eats a cell that ate a cell. And then tertiary endosymbiosis is when a cell eats a cell that ate a cell that ate a cell. Okay, and so that's how you acquire these mixotrophic traits.